Hello, I'm Martin Blaser. I'm the director of the Center for Advanced Biotechnology and Medicine, or CABM. Today, I have the pleasure of speaking with CABM resident faculty member, Dr. Masayori Inouye. He is a professor in the Department of Biochemistry and Molecular Biology at the Robert Wood Johnson Medical School, and he's had a long and illustrious career. Masayori, it's nice to see you today.
Great.
Can you tell me a little about your education and your early career before you came to CABM?
Yeah, I graduated Osaka University. It's 1963, and then I started the postdoc and the Genetic Institute there, working on to determine a genetic code of actual genes. By that time, the all 64 genetic code been deciphered, but we do not know. The genetic code is degenerated, and so which particular codon at each amino acid being used. So for that, we use a T4 phase lysozyme, and we have to determine the purified protein, determine amino acid sequence, as well as double frame shift mutation. So if you have double frame shift mutation, between that mutation, amino acid sequencing being changed. So by comparing wild type mutants, you can determine genetic code unambiguously under that. So this is the first time been done the showing a part of gene sequence, and this is the first time in a way been determining DNA sequence, which is 12 years before Sanger came out sequencing DNA.
Mm-hmm. So it sounds like you were making terrific progress in Japan. What made you decide to come to the United States?
So after my 5 years postdoctoral work in Japan, working on T4 phase lysozyme genetics, I couldn't find a job anywhere in Japan. Decided to go to Princeton's and in Arthur Pardis lab. There I started totally a project on cell divisions and membrane proteins. And that work turned out to be pretty well, and I got the job offer. Let's see, 1970, I have to be careful, from Stony Brook as an associate professor. Yeah. At that time, I was 36. And so comparing with all the American colleagues, I'm pretty old. And so only way I thought I have to work harder than them. I start to keep working on this membrane protein program, and working pretty well.
You went to Stony Brook as associate professor coming from Princeton. And then by the time you left, you were the chair of the Department of Biochemistry.
Right.
And then you came to Rutgers to be the chair of biochemistry here.
Right.
What made you decide to leave Stony Brook and come to Rutgers?
So I came and I met the Aaron Shatkins and other chairs, and I was pretty impressed, and I decided to move here and as a chair. And so I came here 87.
Yeah, so that's more than 30 years ago.
Right. Yeah.
30 years ago. What new project are you working on now that you find particularly exciting?
Well, I started to work on evolution genetic code, how genetic code started at the time of origin of life. And also, I'm sort of ongoing project on toxin, antitoxin system in bacteria, including E. coli, Staphylococcus, and the Bacillus anthracis.
In 2019, you got a very pleasant surprise. You were elected to the National Academy of Sciences. Did you expect that?
Very peculiar to become a member at the age of 85. And, and I'm telling you that to be a member of the National Academy of Science, you have to live long.
If you had to write your scientific obituary, what would you want to be remembered about?
When I was at Princeton, Arthur Pardee told us in a lecture to graduate students how to be successful scientists. He said we ought to change the project every 7 years. So I've been doing that every 5 to 7 years. I'm changing a new project. So I'm working on probably 15, 16 different projects. And so I'm not sure what is, if someone asks me what is my major contribution, my philosophy, whenever I find something unusual, what we're doing experiment, we just work on that. And rather than sticking on the project you've been working on. But recently, the paper show up in Cell, finding a new function of retron, retron, which is consists of some peculiar DNA RNA complex, which been made by reverse transcriptase. And so that is one thing maybe remembered.
Of course, this just appeared in Cell now, but you had discovered it many years earlier, maybe 30.
Yeah, 36 years ago. It's a time where Stony Brook, and we found that.
Yeah,
and we did not know the function of it.
Thank you very much. And arigato gozaimasu.
どういたしまして。
And thank you very much for this interview.